You know, when we're talking about molecular biology, one of the things that always strikes me is how how recently this was discovered. And so, if you ever have the opportunity to go back in time to the early 1960s or late 1950s, a very groovy era, you should bring your lab manual with you. If you just brought your lab manual with you, just this one lab that you are going to be doing about molecular biology, you would earn so many Nobel Prizes for the information that's just in that lab manual. It's crazy. And one of the things that's in the lab manual is the human genetic code. The genetic code, it's right there. This is one chart of the human genetic code. This particular chart for the human genetic code is using messenger RNA codons. That's important to know because DNA codons would be different, right? I could do a chart of DNA codons, it'd be really easy, but the DNA codons would be the complement of this, right? So the DNA codon for this messenger RNA codon would be AAA. You're going to need to remember that. You also need to remember that this is not a transfer RNA codon chart, all right? Now, one of the things you're going to need to do on exam 2 is I'm going to give you some DNA and say, tell me what amino acids this is the code for. Or I'm going to give you messenger RNA. And again, I'm going to say, tell me the amino acid sequence. So you need to be like a ribosome and you need to decode that information. So let me show you how to read this one. Here's the first base, here's the second base, and here's the third base. So let's just imagine that I have given you the DNA, and the DNA is going to be ATC. Oops, it's not. Evidently, it's going to be ACG. Oh, I hope that means something. All right, so now you can't find ACG over there. Well, okay, you can find it, but this is not a DNA codon chart. So if I give you DNA, the first thing you have to do is transcribe the DNA into messenger RNA. So what would the messenger RNA be? It would be UGC. Great. So the first base is U. So that means that the answer is somewhere in here, right? The second base is G. So G, ooh, okay. So the answer is somewhere in there. The third base is C, UGC. That is cysteine. So the answer would be cis, okay? I don't need you to memorize that CYS represents cysteine, okay? The answers are going to be just CYS in a multiple choice. Let's do some more, all right? So if I gave you the DNA TCG, first you got to translate it to AGC. Great, right? Then you're going to go, the first letter is A. All right, the answer is somewhere in there. Terrific. Then the letter G. Okay, the answer is somewhere in there. Why? Because A is the first letter for all of these codons, and G is going to be AG is going to be the first two letters of all of those codons, right? And then you need the letter C, so serine, AGC, right? Now, okay, let's do this one. So ATA transcribes to UAU, terrific. So if I gave you the DNA ATA, you would transcribe it to UAU, right? So take a moment, see if you can figure this out. UAU, do you have it? All right. So U is the first one, A is the second one, and U is the third one. Great. UAU, the answer would be tyrosine. Now, here's something that I see happen to students all the time. If you're listening to this, I'm warning you about it. One of the questions gives you DNA. If I give you DNA, you transcribe it, right? But if I give you messenger RNA, let's say I said, here's the messenger RNA, GCC, you go straight to the chart. Why? Because this is a messenger RNA chart. So if I gave you GCC, you would have to go G and then C and then C. Okay, it's alanine. right? If you made the mistake and you went CGG, then CGG, it'd be arginine. And you'd be like, I know I did it right. I know it's arginine, but you wouldn't find that answer there and you'd think it's a typo in the exam. It's not a typo in the exam. What you did was you found the anticodon for the transfer RNA. This is not a transfer RNA anticodon chart. So if I give you DNA, you transcribe it, then you go to the chart. If I give you messenger RNA, you go right to the chart. All right. So remember my protein. No, my protein is not going to be on the exam, so don't worry about it. But remember that all of these letters mean something, and now you know you could go back and you could look it up, because here, see, so here I've got, here we go. Oh, I did it wrong. GCG means alanine, right? Now you could go figure it out too. One more thing I wanted to tell you about. Now that we're here, I wanted to tell you about a second reason why molecular biology ends up creating such a really, really cool system. The genetic code is described as degenerate, which sounds like a value judgment. I don't know why they use that word degenerate, but here's what they mean by it. If humans would have set up this system, we would have said there's 20 amino acids, there's 64 codons, every amino acid gets 3 codons. So this one, tyrosine, gets 3 codons. Everyone gets exactly 3 codons, right? But that's not the way things happened. The way things happened is life said, you know, there are some amino acids that are so important that if I made a mistake there, if I had what's called a mutation in the DNA, and I made a mistake and got the wrong amino acid there in my protein, protein wouldn't work. Arginine is one of those. Let's look at arginine here and arginine here. All right. With arginine, how many codons are there? There is CGU, that means arginine. There is CGC, that means arginine. CGA means arginine. CGG means arginine. And AGA means arginine. And AGG means arginine. Look at all of those things that mean arginine. Why? Why is that good? Because of genetic mutations. We're going to talk a little bit about mutations. And here's the thing about mutations. There is a type of mutation where there's a mistake in the DNA and the letter C got changed to a G. If there was a change like that in the DNA, so a C got changed to a G, it is definitely a mutation. There is now a mistake. But does it matter? Well, it ends up, no, it doesn't matter. Because CGC means arginine and CGG means arginine. That's like the word gray, that means the color gray. And in British, they spell it gray, right? If I made a mistake and I spelled my gray with an E instead of an A, no one cares. It still means gray. I can actually have a typo in my document and no one cares because the words mean the same. The way the system is set up, there can be 6 different mistakes in the codon for arginine, and you still end up with the right arginine, so no one cares that a mutation has been made. All right. Let's talk a little bit more about how important transcription is, okay? Transcription is going to be the reason that the different cells that we're going to be talking about look the same. If I were to look in the nucleus of this cell up here, this one right here, I look in that nucleus and I compare it to the DNA inside of that nucleus, the DNA is exactly the same. And yet This is a small cell that doesn't move at all, and this is an enormous cell that is one of your muscle cells. Why are they different if they've got the same DNA? They're different because the nucleus of these different cells will transcribe different messenger RNAs, so the cells make different proteins, right? Your eyeball cells do not make any muscle proteins. Your muscle cells do not make any eyeball proteins, right? The muscle cells only transcribe the genes that are going to allow it to make the proteins so it can be the best darn muscle cell possible. It's kind of like there's an entire library here at Cerritos College, and you will find that if you are a development major, you'll spend a lot of time in parts of the library reading all of those books, using all of those books, and you may never go into the accounting part of the library at all. The accounting majors spend a lot of time reading those accounting books, and they never open up a biology book, right? So the different part there, the whole library is the same for every student on campus, but the books that students read will be different depending on their major. These different cells have different majors. They will transcribe different genes, okay? So that is called transcriptional regulation. All the DNA is the same, so all the genes are the same, but the nucleus of a muscle cell will send out different messenger RNA than the nucleus of an eyeball cell. Okay? Let's talk about mutations. You need to know that there are two different types of mutations that I want you to know about. There are mutations that are called point mutations and mutations that are frame shift mutations. The point mutation, it is the substitution of one nucleotide that'll change one codon. When it changes one codon, it might change one amino acid or it might not, right? So let's go back to talking about this, okay? If I changed one nucleotide, if I substituted one nucleotide, so now instead of a U, it is a C, I did change that codon with my mutation, but I didn't change the protein at all. And that is why point mutations, they are mutations, and mutations are permanent changes in DNA. But a mutation that's a point mutation, it might not change the protein that gets made at all. It might actually be completely harmless, right? Frame shift mutations are different. They are the insertion or deletion of a nucleotide. They not only change that codon, but they shuffle all the codons afterwards, and they are always disastrous. Let me give you an example of a frame shift mutation. So I didn't have to do that. Okay. All right, so a pen. So cat ate rat. All right. Cat ate rat. Cat ate rat. Okay. So if I had a point mutation, I would have maybe something like cat ate rat. Okay. Now I've changed one codon, and that did make the sentence hard to understand. However, if we started out with cat ate rat and we ended up with cat ate rat, we might still know what it means, okay? That is a point mutation. Point mutation, it was a substitution, right? Now, let's do a frame shift mutation of the same thing, okay? A frame shift. I am going to insert the letter E, okay? Now I'm going to have this. What happened? All I did was insert an extra nucleotide. But you see how, yeah, it still says cat ate rat in there, But now everything is nonsense. And if we had a whole paragraph that this had happened to, then the entire paragraph got shuffled, right? So a point mutation is the substitution of a single nucleotide, okay? A substitution means I substituted an E for an A. It was a substitution of one nucleotide. But a frame shift mutation is the insertion, adding an extra one, or taking one of them away. And that not only changes that codon, it shuffles all of the rest of the codons after that. Alrighty. The next video, I'm going to actually be talking over a really neat video that I always show in my classes to try and put all this information together. So I hope you watch it.